Another human application of recombinant DNA technology is the production of insulin. Now, we are dealing with biomedical products, and one of that is insulin, which is produced out of recombinant DNA technology process, and we have insulin. Now, what is this insulin? Firstly, it is an endocrine hormone that is produced by pancreas. What is the purpose of insulin? It regulates the blood glucose level. Because you are at that stage of your studies where you have studied the human physiology, you have an idea about what pancreas are, you know where in pancreas this insulin is produced and how it regulates the composition of glucose in the blood. You are very well aware of it. Now, supposedly this part of the human body is affected by some or other circumstance that is persistent. Somebody loses the pancreas, some autoimmune disease comes and starts consuming the beta cells which produce insulin. Beta cells of islet of Langerhans are responsible for production of insulin. So, these beta cells are consumed by some autoimmune mechanism or they are deteriorating. So, we have a condition known as diabetes mellitus. This diabetes mellitus is of two types, type 1 and type 2, while type 1 is autoimmune, the other one is not autoimmune, and the last condition is that the pancreas have been removed. Now, this diabetes mellitus is a disease that is growing day by day in human population, and how do we control it? We control it by administering insulin externally to the patient. Now, the insulin, as it is not being produced, this is gone, no insulin production would be there. When there would be no insulin production, this blood glucose would increase and the patient will suffer from this condition. Now, how to control it? You cannot bring back this, okay? You have to bring this down. So, what you can do is externally provide insulin, okay? Now, externally providing insulin was practiced till quite lately, till 1990s, when the recombinant insulin came into the scenario, it was replaced. Now, what was the problem in the previous administration of insulin that we had to switch to the recombinant insulin? The problem was that it was extracted from the cattle pancreas or the gut of the cattle. Now, you know, if the insulin is extracted from cattle and then given it to the human beings, there is an advanced immune system going on inside our body. It is working continuously inside our body. Something from outside is coming inside the body. The immune responses are bound to happen. So, in most of the cases, the patient who was administered the insulin from a definite cattle, he or she was bound to give an immune response. So, in order to overcome that, with the advent of rDNA technology, it was decided or it was perceived that if we give the insulin that is produced in the lab by a transformed host, it would be very easy for the patients to get this insulin. Now, how it is carried out? You know, insulin is present inside the body or it is formed inside the body as pre-proinsulin and later on it gets converted to proinsulin. Before this, it is pre-proinsulin. Okay, pre-proinsulin is transported to endoplasmic reticulum. Over there it gets converted to proinsulin, and this proinsulin later on gets converted to insulin. Now, what happens is, like, this proinsulin has 3 peptides. It is having A peptide, B peptide and C peptide. Out of the 2 peptides, A peptide and B peptide are necessary, while the C peptide has to be removed. Now, pay attention, this is A, this is B and this is C peptide. Now, what is to be done is that this is A, this is B and this is C peptide. Now, C peptide is to be removed and then it becomes insulin, and A and B have to be attached by disulfide bridges. Now, the conversion takes place in such a way that you have A peptide, you have B peptide and you have C peptide gone away. It's cut. A and B, they have disulfide bridges in between and that forms the insulin that would be responsible for the regulation of blood glucose level inside your body. This is A, this is B, this is C. A peptide, B peptide, C peptide has been removed. There are disulfide bridges present. Now, this is insulin. This is proinsulin. Now, what is to be done in recombinant technology is that this insulin has to be synthesized inside the E. coli. A company known as Eli Lilly in USA is responsible or has the feather in its cap for first of all making insulin, commercial insulin, where they used E. coli. Inside it they had the vector plasmid carrying the gene for formation of insulin and that gave rise to insulin. Now, here we have same thing, this is the E. coli host, this is its nucleoid, this is the vector DNA. Vector DNA has the gene, insulin gene. So, green part represents insulin gene from a healthy individual is incorporated inside E. coli and this E. coli produces insulin when the translation takes place. So, we have insulin being produced that is sold commercially. Okay. Now, it is so easy to produce insulin, you can produce as much amount of insulin as you require. You need not to put any burden on the environment by killing the cattle, enormous amount of cattle. There is no immune response that would be generated when we give such an insulin to the patient. So, you get an idea, this is another important human health application of the rDNA technology, where the principles of recombinant DNA technology are used to produce a biomedical product that can be utilized for covering a covering a disease which has a huge coverage in human population, and that is diabetes. How that is covered? That is covered by producing commercially insulin using rDNA technology, where the transformant host is producing the insulin. There is no amount of tension that is present where we just assume that there could be an immune response from the patient. Instead, such an insulin is very much user friendly. It is very user friendly. It could be injected inside the body directly. And do keep in mind that such protein cannot be put into tablets and administered orally, because insulin is a peptide hormone and it gets digested in the stomach. Our stomach is very well able to digest the proteins. The protein digestion starts taking place inside the stomach itself. So, we cannot use this insulin that is being produced, we cannot administer it orally. We have to inject it into the bloodstream itself, so that the digestion does not take place in the stomach. Now, you have to keep in mind the plus points of this particular recombinant DNA technology using produced insulin, and I hope you get an idea how important this particular process can be if we have a good aim of improving the human health conditions which are often genetic, and not only genetic cases. As we saw in the gene therapy, we are seeing that many biomedical products can be produced using this particular technology.